So we're out in the woods today. Hi, my name is Paul, and welcome. Today we're going to be doing a little bit of experiment. We're out in the woods. We're going to be exploring the microbiota of the forest. So one of the techniques that we're going to be using is rice culture. So we have rice that we cooked partially, and you can see it's not mushy, the seed and the grains of the rice, but you can still break them apart. So it's only partially, like half cooked. And the ratio that we're using is about 6 cups of rice to about 4 cups of water. So you're not fully cooking the rice. And what we're going to be doing is going out and using this rice. We're going to collect some samples of soil randomly in the forest. This is a healthy forest. So we're hoping that what's in here is a healthy microbiome. And so what we want to do is take a look at what the different microbiomes are in a bunch of different healthy forests and see, okay, what fungus are we getting? What bacteria are we getting? And then we're going to collect that sample and we're going to preserve it, and we're going to bring it back home, and we're going to use it to inoculate some more soil. And so we're trying to bring in healthy microorganisms, a healthy microbiome into our garden. Because when we do gardening normally, we're tilling the soil and we're wrecking the soil, and we're disturbing all this microbial community that needs soil that's intact, that's not being constantly broken up, because the fungus are long hyphae and they grow out into the soil, and every time you cut into that, you're breaking it. And so over time, again and again and again, you're decreasing the amount of fungus in the soil, and all these beneficial microorganisms that are there are decreasing. And so what we need to do when we're doing a regenerative agriculture system, or if we're restoring an ecosystem, is we need to bring all those healthy microorganisms back into the soil. So they're doing all sorts of really great stuff. And for the plants, they're actually bringing in, when they have an association with the plant roots, they bring in a lot of water. So the roots only go out so far, but then the hyphae of the fungus goes even farther out, and they can collect phosphorus and all sorts of micronutrients, and they bring it into the plant. It goes up, and the plant is nice and healthy, and it's protected from microorganisms that are pathogens. And also the plant brings that nutrient down into the soil. It photosynthesizes, it produces sugars, and the sugars go down into the soil, and they feed the fungus and the different bacteria that's in the soil. And so it's a symbiotic relationship. So they're both benefiting by having this movement of soil nutrients up into the plant and sugars down into the soil. And the fungus can actually manipulate the plants a little bit by having different hormones and different things that stimulate the plants to produce more roots and produce more sugar for them. So it's a very interesting thing. And also these hyphae that go out into the soil, they can go for such long distances, and they actually help with communication between the different trees, and it's what we call the wood wide web. All these different organisms are actually all connected. You think they're unique, but in a healthy, intact ecosystem, they're actually communicating with each other all the time and creating a much more resilient and healthy ecosystem. So that's what we're trying to do. We're doing a little bit of experiment. We're going to see what we can find in a healthy forest, and we're going to bring that back, and hopefully we'll be able to transfer those bacteria and fungus into our own soil, and then see how that works with growing our own plants. So there are many different ways that we can collect the microorganisms from the forest. This is one way. We're using the rice, and so it's basically a trap. So we're putting it out into the forest and allowing all the microorganisms to land on it, and then they'll start growing and expanding their hyphae network. And then we want to take that and bring it back into our nursery, and then we're going to be teaching you how to propagate this and expand it out so that you're getting more and more microorganisms so that you can spread it over a much larger area. And so if you're doing like a large-scale ecological restoration, or you're doing a large regenerative agriculture project, so that you can use these microorganisms in your project and expand them out and have them yourself. So just trying to show you some simple techniques to do that, and I hope you join me and enjoy these different experiments that we're doing, trying to figure this out ourselves. So here I have my handy Ziploc and a shovel. And what I'm going to do, this is a nice big old tree, so I know it hasn't been disturbed for a very long time. So just coming down here, you can see very organic soil. So really don't need very much, because within just a little bit of soil like that, you've got thousands, millions of different microorganisms in there. And so if we're just taking a little sample, and then we'll just cover this back up again so we're not harming the site. And then we'll just keep repeating that. We'll do about 3 or 4 different samples in different spots, because we find that soil microorganisms, if you're sampling within the same spot, you're not going to get a lot of variation. But if you move around a little bit and you get a little bit of soil here and a little bit of soil there, you're actually getting more diversity of microorganisms by doing that throughout the site. So here I've got my little soil sample, and I just mixed it up. I'm just going to take a little bit of this. So because this has millions of microorganisms in it, I'm just sprinkling a little bit on the top here. That's probably enough, I think, just to inoculate. And we'll see what develops from that. In a couple days, it'll be covered with fungal hyphae. So here I'm using rice, but I mean, you can use any sort of carbohydrate. It's just an energy source for the fungus so that it can really grow really quickly. And it provides a substrate for them to grow into and grow rapidly. So you can use oats, you can use any sort of grain, you can use cassava, you can use potatoes even probably. Just something that doesn't inhibit the bacteria and the fungus. So here you can see I've inoculated these 5 different containers, and I just sprinkled a little bit of the soil. We collected it from 3 or 4 different spots in a healthy forest. So then I just mixed the soil up to mix the microbiota around a little bit, and then sprinkled a handful on top of the rice. And I'm curious to see what sort of microbial communities grow out of these different sites. I think I'm pretty excited about that. So what we're seeing here is the fungal hyphae, and it's white or cream, which would be sort of Bavaria, and it's an endophytic fungus, and so that means it colonizes the outside of the roots and forms sort of a protective barrier around the outside, and that's beneficial, and it has a symbiotic relationship with the plants, helping them in many different ways. And some Bavaria are actually parasites on different insects, so having them in the soil helps protect against plant or for different insects as well. On the side, we're seeing yellow, and that's more of a bacillus. They colonize plant roots and they attack plant pathogens. Think of them sort of like a shield for your roots. The green is more trichoderma, and it colonizes the plant roots, and it has many benefits as it improves plant growth, helps with mineral uptake, and it inhibits plant pathogens. So that's all good stuff. We don't see any red or pink, which would be more fusarium, which is a pathogen, and we don't see any purple or lilac, which is rhizoctonia. And so those pathogens we don't want in there, and we don't see any in there. So it's a very healthy soil that we're getting from these healthy habitats. These different pathogens can cause root rot and damping off, so we don't want them in there. The blue mold on the top is more penicillium or aspergillus, and both are beneficial to plants for growth and for promotion of growth and for protection from different pathogens. So they're both excellent to have in there as well. So it's really nice when what we're doing here is we're just looking at the different species that are within the soil samples, but you don't really need very much soil at all. And then you can put them into your medium, you can put them into your superbrew, you can propagate them by rice culture, or you can use an aqueous solution as well of starch, and just propagating them out just by using like a teaspoon. In a teaspoon, there's millions of different fungal spores and fungal hyphae, and bacteria as well. So you really don't need very much you don't need to harvest a lot from the forest to do this. And you're looking at healthy ecosystems, and we're bringing it into our garden. And once they're in there, they're going to stay as long as you do good cultural practices. You're doing no-till, you're mulching, you're feeding the bacteria with the superbrew, you're not disturbing the root zone very much. You're doing chopping the plants off and just letting them drop rather than ripping them out of the soil. Different things to keep that microbial balance in the soil and promote it so that you have the healthiest plants. The bottom half of our collection, you can see, is still full of microorganisms, and we're going to mix that up. And we've got 5 different sites, and you can see there's a little bit of dirt in there still, but it's rich, very rich with microorganisms. And we're going to take that and we're going to put it into a bucket, and then we're going to mix it up with some water, and then we'll put it into our biological fertilizer, our superbrew. And here you can see it's in the bucket, and we're just breaking it up. And you can see all the different colors in there, so it's got a good diversity of different species. It's really rich, and that's what you really want, that whole diversity of species from the forest. And here you can see we've got our superbrew, and we're putting our microorganisms in, and I'm just going to put about half of that mixture in there. And then the other half I'm going to spread on the garden, fill it back up with water, and just spread it around the garden. And then the superbrew we can use all summer long, and it'll keep giving us a little shot of microorganisms throughout the season. So just having that brew and having all the micronutrients and all the fungus and bacteria in there is just like so rich for your soil. It's amazing. And here you can see we've got a bag of sugar, and we've got our 5 different sites, and put them into the bowl, and you can see it's rich with fungus. And yeah, so we're just taking that, we've got soil in there as well, so that's the dark stuff, and the leaves, and mixing that with 50% sugar, and that lowers the osmotic concentration, and it stops the biological activity, and so the fungus cannot grow anymore. So it's just kind of putting it in stasis for a while, kind of the same process that you're using for jam. And then we'll just mix this all up, and we can put this into a jar, and we'll put a breathable cover on top of that so that it can still breathe because it is still alive, but the biological activity is very low. And we'll put that into the refrigerator or into a cool, dark place, and we can use that, adding a little bit into our water solution or supplementing into our superbrew again, to make more biologically active throughout the summer. Just giving a little bit of a boost here and there just gives it a little bit of a charge. And here you can see how it looks when it's all finally prepared, ready to go into the fridge. And that should last for many months, being preserved like that.